I get anxiety when I can't sit there. Hey Mo,
Hey.
can you send me those files too?
Which files?
The ones that I need to ID.
Okay.
Yeah. I just need to look at what features we want to fragment.
Okay. I'm always the cameraman. Yeah.
I sit next to you now.
Yeah.
Thank you. Real sharp. Yeah. All right.
So I was able to send it for this.
Ryan, thanks for sending that note.
I know. I'm gonna need his money. So I'm
Yeah, watch it.
Sounds good. Let's hope.
I heard you will.
How are you?
No, okay. Let's just give me.
Yeah. Yeah, so,
You know, I think mine is my favorite juice.
I agree.
Okay. Because mine's something that doesn't.
Yeah.
Yeah, I think
Good luck, Luke.
Take one.
Action. It's going. It's going.
Relax. No?
Why don't you just open all the lights? Come on.
What?
Do you turn on the lights? It's dark. It's kind of dark.
No, I think it's fine. This is what'll keep the lights out.
Remember your visit.
You want to say hi?
Shout out. Shout out. Period in the statement. Family, friends.
Wow. Is this a funeral or? I don't know. I always get a little worried when people are so well dressed up, and we look so crummy, right? Good.
I mean, I thought about wearing, my new
No,
jeans,
you look
t-shirt,
great.
and a hoodie, but I thought this was more
You
fitting
look,
and church.
yeah, you look great. You look great. That's good. We should dress up more often.
Yeah.
I agree.
I heard right now they're having a really big train migration.
Hey, Mariah, quick question.
Yeah.
All right. So I am on Notability.
Okay.
Pum, 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 pum. Oh, no. Let's say I No, let's see. I want to. What if I wanted to insert a page?
Yeah.
How do I do that?
So you go here, and then so you do this, add page, a blank page in between, so you can go here and write your notes about
Wait,
this
wait.
page.
How did you do that?
Yeah. So you go. Okay. Let me delete it first. So you go to this, and then.
Right. So okay,
yeah,
got
so
it.
I will cut this page. No, don't worry.
Okay, so let me try it again.
Yeah.
So you do this. Add page. Wow.
Yeah.
Super duper.
Yeah.
Thank you.
And
That's what I
then
was missing.
something. Let's say you have a super long paper and you wanna search one word.
Definitely.
Let's
Right.
say SBC, or SP whatever, SPO. So SPO present in all of these papers that listed now in the index.
Ah,
Yeah,
cool.
yeah, and yeah. So these
Yeah,
are
yeah,
all.
yeah,
And then
cool.
remove it, and then go back to the entire thing. Yeah.
Super duper.
Yeah,
Thank you.
yeah.
That's what I was.
I see you reading it.
I'm reading it.
Give everybody a chance to read it.
All right, I didn't get it.
Wait, we're missing.
I didn't. Was I supposed to know?
Who are we missing? Martin.
Yeah, he needs one more, right? It's Jen, right? No?
Jen,
Yep.
Valerie, and then me and Martin. Okay.
Wow, this is so cool.
You learned a new trick.
Yeah, how to insert pages. Mohammed, you can't graduate, but you can only graduate if I finish learning how to use Notability.
Yeah.
Is he wrong?
Learn how to use what?
Okay.
Learn how to use what?
Notability.
Notability.
What's that?
That's a program for taking notes on iPad.
It's actually what I used in the seminar. Read the talk. It's also, but you can insert PDFs
Notability?
and pictures and yeah.
Notability, yeah. Only Mohammed. So Mohammed, did you hear what I said? You can't graduate until I know everything
Okay,
there is to know
I'm
about
fine.
Notability.
We can have an intensive
Because
course.
otherwise I'm going to be lost.
This probably wouldn't be a short course. It's more of a long course.
Yeah, we're slow learners, you know, Brian and I. Yeah.
That's the problem with faculty.
Yeah, first of all, they don't listen, and they don't remember anything. So,
Who's that?
us.
Faculty. All right, well, welcome everybody. And thank you for joining us here this morning for Luke Berry's PhD defense. So I'd like to start off, so I'm Brian Bothner, his advisor. And I'd like to start off with just a little background on Luke. So he came to Montana and MSU from Maryland. So he did his undergraduate work at Hood College in Frederick, Maryland. And you know, Luke even admits this, that he came to Montana for the mountains. And then he stayed for the research. And you know, we're often suspect of these graduate students who come for the mountains because sometimes they don't turn out to be the best graduate students. They can be great mountain climbers and skiers. But Luke has turned out to be one that he came for the mountains and he really got into the research. And he really took advantage of, you know, all of the resources here and really applied himself and has done some really impressive work using mass spectrometry to understand protein structure and function. And you know, my group has been doing this for a number of years, and I think in some ways that can be a problem for graduate students because every student is expected to raise the bar, right? You've got to always one-up the graduate student who you're following in the footsteps of. And Luke had some pretty big footsteps to fill. You know, the other students really put the bar pretty high for doing biophysical analyses using mass spec. You know, Ravi Kant, Naveed Movahed, Jonathan Hilmer, Vamsi, Raya Prolu. But Luke has really done that. And he has pulled off sets of experiments with hydrogen deuterium exchange that I can just flat out say no one else in the world has done work at this level. And, you know, working, for example, with oxygen-sensitive proteins, which those of you who work with these, like people in the Dubois group, you know, oh my gosh, the level of complexity this adds to every experimental step. So it really takes someone who has impeccable technique to pull this off. And so, you know, Luke has done this a number of times throughout his career and has published, you know, some really wonderful papers to really push the edge of what you can do and also really help us understand how, you know, protein structure and function are related. So with that, Luke, I'm handing it over to you. And
Thank you so much, Brian. And I will say, I'm not that great of a skier and a subpar mountain climber. So the title of my talk today is The Role of Protein Dynamics in Electron Bifurcation. And I'm really excited to share all of the really cool stuff I've been able to find out about these unique protein systems. But first, to start off, I want to give a little background on electron transfers and how they relate to the generation of usable energy in cells. So electron transfer can be broken down into an oxidation and reduction reaction. In an oxidation reaction, we have a reduced compound, in this case A, that loses its electrons, becoming oxidized in the process. And then the other side of that is the reduction reaction, where an oxidized compound will gain those electrons, becoming reduced in the process. And so the tendency of a chemical species to accept or donate an electron or to become oxidized or reduced is determined by its reduction potential. And so lower potentials indicate that it's much more likely to donate an electron, and higher potentials indicate that it's much more likely to accept. And so cellular respiration is the main source of energy for cells. And one I'm sure everybody here is familiar with from high school when we first learned mitochondria is the powerhouse of the cell. And the overall reaction for cellular respiration is the oxidation of glucose to carbon dioxide and the reduction of oxygen to water, all while producing ATP for the cell. And so the ATP acts as the energy currency in the cell, and it's because the hydrolysis of ATP is a very exergonic process, releasing a lot of energy. And so that energy can then be harnessed by more endergonic reactions in order to drive them forward. And so there are two primary mechanisms of energy conservation. The first is substrate level phosphorylation, which includes glycolysis and the citric acid cycle. And both of these produce small amounts of ATP when their main function is to produce electrons in the form of NADH and FADH2. And these electrons then go on to the next mechanism, oxidative phosphorylation, which includes the electron transport chain and chemiosmosis, which produces the bulk of the ATP available in the cell. So something interesting about the electron transport chain is that there's this unique mechanism of electron transfer known as electron bifurcation. And so this was first discovered in complex 3, which I've shown pictured here. And the process is called the Q cycle. What the Q cycle does is it uses the oxidation of ubiquinol in order to reduce cytochrome C, another equivalent of ubiquinone to ubiquinol, and then transport protons across the intermembrane space of the mitochondria. And so the role of electron bifurcation in complex 3 is to increase the efficiency of generating this proton mode of force while minimizing the changes to the overall electron flow and minimizing energy loss to the system. And it does this by coupling this favorable electron transfer, this high potential first transfer here from the Q0 cofactor to the iron sulfur cluster, with the second low potential transfer from Q0 to the BLBH hemes to reduce ubiquinol. So to kind of give a little more simplified version and explanation of electron bifurcation, we have a spontaneous downhill electron transfer and a non-spontaneous uphill electron transfer. And so these systems couple these together by using the energy provided from the spontaneous electron transfer to drive the non-spontaneous electron transfer without having to do any additional input. And so the reason, so electron bifurcation is getting a lot more and more attention because it's being implicated to play an important role in anaerobic metabolism. So anaerobic organisms do not have access to the large amounts of ATP that we do. So as an alternative, they use the oxidation of ferredoxin and flavidoxin, these monomeric proteins, to provide energy for these inorganic reactions to proceed. However, the reduction of ferredoxin and flavidoxin is extremely difficult individually. So these organisms have adopted electron bifurcation, and as an example, by coupling the oxidation of NADPH and the reduction of NAD in order to produce ferredoxin. So the way this works is that NADPH will donate 2 electrons to the central flavin molecule that acts as the bifurcating center. The 2x reduced flavin will then spontaneously send an electron down the exergonic branch to reduce NAD to NADH. And then the remaining 1x reduced flavin has a sufficiently low reduction potential to drive the endergonic branch of reduction to ferredoxin spontaneously. And so since the discovery of electron bifurcation and anaerobic metabolism, there have been several enzymes characterized and identified to have this mechanism. And these include the NADH-dependent ferredoxin NADP plus oxidoreductase, which is always a mouthful to say, so NFN for short. And there's also the electron transfer flavor protein, or ETF, and the iron-only hydrogenase, or HYDE. So what's unique about these 3 enzymes specifically is that they all function in order to generate reduced equivalents of ferredoxin. However, the overall bifurcating mechanism and the source of the electrons for it is quite different. So for instance, in NFN, we have a heterodimer with 2 flavin-adenine dinucleotides, a 2-iron 2-sulfur cluster, and 2 4-iron 4-sulfur clusters with NADPH as the electron donor. In ETF, we have 3 flavidoxins, or no, not flavidoxins, flavin-adenine dinucleotides, 2 4-iron 4-sulfur clusters. It uses NADH as the electron donor, and ubiquinone as the exergonic electron receptor. And then finally, with the hydrogenase, we have 4 2-iron 2-sulfur clusters, 6 4-iron 4-sulfur clusters, and a single flavin mononucleotide. And it uses hydrogen as the electron donor to this unique iron-sulfur cluster called the H cluster, which also acts as the site of bifurcation and hydrogenase. And so the bulk of my, so really the first part of my research was focused on characterizing the structural dynamics of the NFN protein. So I'll give a little more background on that. It's a heterodimer complex with this small subunit in green called NFNS, large subunit in blue called NFNL, and then it contains 5 cofactors for bifurcation. This 2-iron 2-sulfur cluster here on the small subunit, 2 flavin-adenine dinucleotides, one in the small subunit designated as SFAD, one of the large subunit designated as LFAD, and then 2 4-iron 4-sulfur clusters designated as proximal and distal relative to
Can I ask a
distance
quick
level.
question? Sorry. Where does the NADPH come from? Is it pentose phosphate? Or?
I don't know specifically if that's the pathway it comes from, but it's abundant in this, it's just present in the cell. And then,
Sorry to interrupt.
no, you're fine. So this catalyzes, as I mentioned on the left side, the oxidation of NADPH to reduce NAD and ferredoxin. And then also, a unique or an interesting fact about this is that NFN can also do the conforcation of the reverse reaction. So it can oxidize ferredoxin and NADH in order to produce NADPH. And this is thought to help kind of maintain the balance of NAD, NADH, and NADPH within the cell. And so right around the time I started working with these systems, there was a study coming out looking at ligand binding and conformational dynamics in NFN. And so the way the demo group looked at this was through hydrogen deuterium exchange coupled to mass spectrometry, or HDXMS. And they looked at different ligands bound or substrates bound to the NFN complex in order to, and they set out to find the site of NADPH binding. But what they also found is that they also found evidence of a rigid body movement in the small subunit that controls electron flow down the exergonic half. And so if you look at the structure here, we can see this green one conformation of the small subunit, and then we have this grayed out secondary conformation of the small subunit. And what this does is it alters the distance between the 2-iron 2-sulfur cluster and the LFAD in order to gate the electrons to prevent multiple exergonic transfers down the exergonic branch. And so this really suggests a potential allosteric mechanism to regulate electron bifurcation in the NFN system, mainly because when we see, this behavior was observed when the different substrates were down. So this really interested me, and I wanted to pursue this further. And so I set up a similar experiment looking at essentially all of the different steps of the bifurcating and conforcation mechanism. So the way I did this was, so the first condition is as purified NFN. So it's just NFN as we get it with cofactors found, no substrates were added. And then we have the bifurcating reaction, NADPH, NADPH and NAD, as well as oxidized ferredoxin. And we also looked at the conforcating reaction with NADP, NADH, and then both NADH and NADP. So, before I get too much into the data, I'd like to just kind of introduce everybody to hydrogen deuterium exchange, so we're all on the same page. The way these experiments work is you start with protein of interest, where the amide hydrogens will freely exchange, of the amide hydrogens of the peptide backbone, will freely exchange with hydrogens in solution. And so we can replace the solvent system with deuterium oxide, which will then cause those amide hydrogens to exchange with deuterium instead, increasing the mass. And so the rate of exchange is heavily influenced by the protein structure. And so more accessible regions, such as on the surface of the protein, will exchange much more rapidly, and more structured regions on the interior of the protein will exchange at a much slower rate. And additionally, we can also use this to look at protein-protein interactions or protein-ligand interactions to identify the site of binding, and then also look at global changes in the protein complex. And then finally, we just quench and digest to stop the exchange reaction and localize the deuterium incorporation, and then analyze it with mass spectrometry. And so for NFN, as Brian mentioned, these are very oxygen-sensitive proteins. So in order to keep them anaerobic, to get as much mechanistically relevant information as possible, I use this 2-vial system with degas buffer, so I can easily access the sample to remove aliquots, to quench and digest, and then freeze right before LC-MS analysis. And so after data collection, we can compare the mass spectra of the non-deuterated and partially deuterated samples, and based on the shift in the isotope distribution, we can determine how much deuterium is incorporated. From there, we can plot the number of deuterium over time to look at how much deuterium is incorporated in each condition over time. And then finally, we can also map this onto a structure of the protein in order to determine where these differences in exchange are happening and relate it back to the function. So the first step in looking at the NFN system, I wanted to see if we could find evidence for some long-distance communication based on differing deuterium incorporation amounts with the nucleotide bound, or with the nucleotide substrates bound. So what I have here is the structure of NFN, and the red highlighted regions are just regions of interest I decided to show you all today. And the histogram shows the number of deuterium incorporated after 3 hours for each of the conditions. And so starting here at this NFNS, a small subunit, residues 131 and 139, which is part of the NAD and NADH binding domain, we see that the biggest decrease in exchange is when NADH and NAD are bound, the orange and green traces. Excuse me. And so this kind of fits our expectations of when one of these substrates bind to this region on the small subunit, we're going to see a local stabilization event, which is going to decrease the accessibility for exchange. And then what we also saw is that when NADPH is present and NADP, we also see a decrease in exchange in this region, which piqued my curiosity because NADP and NADPH are bind down here by the large subunit. And so this starts to tell us when one of these substrates are bound here is causing a slight conformational change here, decreasing the accessibility to exchange. This kind of tells us that maybe there's some relationship going on with ligand binding affinity in order to ensure the bifurcation reaction proceeds. And so investigating this region next, we saw a similar pattern of decrease. However, we saw the sharpest decrease when ferredoxin is present. And so ferredoxin binds down here near the distal 4-4 sulfur cluster. So it's likely there's some interactions going on with this loop region stabilizing it and preventing the exchange. But we also see a large decrease when NADPH is bound, and then also a slight decrease when NAD is bound. And then we also see a very substantial decrease, similar to the ferredoxin, when both NADPH and NAD are bound. And so this, again, kind of helps us confirm that there's some long-distance communication. So when a substrate binds up here, or even down here with the ferredoxin, there's some global changes going on in the conformation of the protein. And then finally, we can look at the cofactor binding site. For instance, here, this red region right here highlights the LFAD binding domain. And we see the sharpest decrease when NADPH and NAD are bound. This essentially tells us when the nucleotide substrates needed for electron bifurcation are present, the main cofactor for the bifurcating center is essentially being aligned to send the electrons down the respective branches. So I thought this was really cool. We could see that there's some evidence of long-distance communication going on through the complex, but we wanted to know specifically how this signal is transmitted. And so to do that, we brought in Saroj from Eric Boyd's group here at Montana State University, who had a lot of experience doing statistical coupling analysis. And so statistical coupling analysis can identify co-evolving amino acid residues in the protein. And it does this by using a multiple sequence alignment of, in this case, NFN-like proteins. And it'll search each amino acid position in that sequence for mutation. So for instance, in sequence 1, it searched, it found a rotation or mutation at this arginine position, and then it looks at every other position in the amino acid sequence, right? Every other position in the peptide sequence or protein sequence to see if it can find another mutation. And if it does, it marks those as co-evolving. So when a mutation occurs at one of these positions, it also causes another change in the other. So the most common example of this is with this arginine, and I'll kick myself because I already forgot, aspartic acid. And so arginine switches with glutamic acid, switching that positive for a negative charge. And so then the aspartic acid has to switch accordingly from a negative to a positive charge in order to keep those charge interactions neutralized. And so with this, we were able to identify several co-evolving residues in the NFN complex. And we actually looked at these in the complex itself. You can see them in this light pink color throughout the complex. We can essentially subtract by the bifurcating LFAD and then the 2 iron 2 sulfur cluster in SFAD of the small signal. So this was really cool. Now we know there's some long distance communication going on. We also know there's some potential pathways of communication throughout the complex. But now we wanted to take it a step further and go, what does the communication do? And so the way we did that was by essentially combining the statistical coupling analysis and hydrogen deuterium exchange data. And so we combined the co-evolving residue, the co-evolving residues identified with SEA, as well as correlation of the HDX-MS data of the bifurcating substrate conditions relative to the S purified. So the network we generated, you see here, each of the nodes represent a peptide identified with hydrogen deuterium exchange. Red nodes indicate peptides with at least one co-evolving residue, and then green nodes represent peptides with no co-evolving residues. And then the edges connecting each of these nodes indicate the correlation or anti-correlation of the deuterium exchange data. So for instance, red edges between this S260 and L245 suggest that they exchange at a similar rate relative to the S purified condition when the bifurcating substrates are bound. So NADPH, NAD, and ferredoxin. So we can actually divide this network into 2 subnetworks. First one here, outlined in red. We can then take the co-evolving peptides onto the subnetwork and map them onto the structure. And what we see is a large density of these peptides around the 4 iron 4 sulfur clusters, the LFAD, and then also at the NFNS around the SFAD and the large and small subunit interface. And so looking at the actual correlation, we see that the region around the LFAD is highly correlated with the 4 iron 4 sulfur clusters and this interface between the small and large units. So that tells us when some of the bifurcating nucleotides are bound, these regions are undergoing coordinated conformational changes. And so looking at the second network, excuse me, looking at the second network, outlined here in purple, we can again map these onto the actual structure, and we see that there's still a large density around the LFAD, and more density around of these network peptides around the NAD and NADH binding site, as well as the SFAD and 2 iron 2 sulfur cluster. This kind of helps confirm the original hypothesis of protein conformational changes were regulating electron gating. This is because if we think about it, between the NAD, SFAD, and LFAD, all of these are undergoing coordinated movements during the bifurcating reaction. So when NAD is bound, all of these conformational changes occur to essentially get the exergonic branch lined up. And then in theory, if we look at the conformating reaction specifically when NADH is bound, we should be able to see a similar one indicating that, again, these are communicating and moving out the distance. I have looked at the conformating network before I can show that here today, because I've got much more fun stuff to show you. So to kind of wrap up and summarize all the work I did with NFN, We used HDXMS to look for evidence of allosteric communication, and then we used statistical coupling analysis to identify the residues potentially involved in that communication. And then by combining them, we formed a network plot that shows regions of NFN undergoing coordinated motions with one another. And specifically, that revealed the electron gating mechanism is controlled by multiple subtle protein conformations instead of one big rigid body movement between the small and large subunits. And finally, looking at the protein motions, that change the distance between the 2 iron and 2 sulfur cluster in SFAD. It regulates the accessibility of the exergonic branch for bifurcation, or for electron transfer. And looking at the reduction of NAD to NADH can cause a conformational change at this region, effectively removing the 2 iron 2 sulfur cluster out of electron transfer distance. And so this really inspired me to look more into these proteins involved in anaerobic metabolism, and to go take it to the next level and look at much bigger complexes. And so the next step I did was investigate the nitrogenase systems. And so the nitrogenases reduce atmospheric nitrogen to ammonia. And the way they do this is through a 2-component system featuring the MoFi protein, shown here on the right, and the iron protein, shown here in the middle, as well as an electron carrier flavodoxin, which, remember to the earlier slide, is synonymous to ferredoxin. And so the way these 3 proteins work together is that reduced flavodoxin will transfer an electron to an ATP-bound iron protein. This iron protein will then transiently interact with one half of the molybdenum iron protein, transfer an electron to the P cluster, and then to the iron molybdenum cofactor, which is the site of nitrogen reduction. So the process of these transient interactions between iron protein and Phi protein and flavodoxin is known as the iron protein cycle. So to kind of take you more through a step-by-step of what the iron protein cycle does. So we start off with one half of our MoFi protein and our reduced ATP-bound iron protein. Iron protein will transiently interact with the MoFi protein, causing the first electron transfer from the P cluster to the Phi MoCo, which then allows the iron protein to transfer an electron back to the P cluster. After electron transfer, ATP is hydrolyzed, causing a conformational change in the iron protein that decreases the interactions between the MoFi protein and the iron protein. And then the next step after that is the inorganic phosphate is released from the iron protein, allowing iron protein to dissociate and start undergoing regeneration. And so to regenerate, the oxidized ADP-bound iron protein interacts with reduced flavodoxin. Flavodoxin will transfer an electron to the iron protein, and then iron protein will then exchange the ADP molecules for ATP, allowing it to repeat the iron protein cycle. And so this has to happen a total of 8 times in order for the nitrogenases to produce ammonium and hydrogen. 2 ammonia and 1 hydrogen gas. So anyway, I was really interested in these because I really wanted to understand the structure-function relationship between the nitrogenase systems, electron transfer, and how that helps coordinate and prepare the system for nitrogen production. And so in order to do that, we need to understand all 3 of the different nitrogenase systems that are available to cells. So the first is this mode-dependent, which uses the molybdenum iron protein as its catalytic component, which I just showed. There's also the vanadium-dependent, which uses the vanadium iron protein, which I've shown here on the right. And there's also the iron-dependent, or the iron-only protein. And so the big difference between these 3 is the active site cofactor uses a different transition code. So for the MoFi protein, it uses molybdenum. For vanadium, for the vanadium iron, it uses vanadium. For the iron only, it uses iron. And so additionally, looking at the crystal structures of the MoFi protein and the vanadium iron protein, we can see that they all have very similar structural homology. Similar structural homology. However, there's this additional gamma subunit shown here in green in the vanadium iron protein. And we know it's there, but it's really not known what it does. Thankfully, it's also present in the iron-only protein. However, there currently is not a crystal structure available for the iron-only protein. So, the first logical step in really characterizing all 3 of these different nitrogenases to determine the structure-function relationship was to develop an iron-only protein homology model. And so we accomplished that by using the Fire2 protein fold recognition server and validating it with chemical cross-linking. So chemical cross-linking will covalently link 2 amino acid residue side chains, and based on the distance of the cross-linker, you can determine their spatial relationship in solution and relate that back to a homology model. So on the left here, we have a cross-linking map generated by Monica from the Bachner group, showing the alpha, beta, and gamma subunits, shown here in the same color on our homology model of iron protein, of the iron-only protein. And so each line connecting the subunit the red specific between the alpha subunit and the gamma subunit. And so we can then map those crosslinkers onto the actual homology model and see that, again, the red lines represent crosslinks between the alpha and gamma subunits. So it's really helped us confirm that the overall structure of our homology model is fairly accurate, and that we could use this as a reference point for some more detailed solution studies. So the next step was to compare the structures of the iron-only protein and the molybdenum iron protein. And the way I did this was using HDXMS, which by the end of this talk you will hopefully know, if nothing else, is my absolute favorite technique. But so the way it works is we compared percent deuterium incorporated after 3 hours in both the MoFi protein and the iron-only protein. We can then map that difference onto the homology model of the iron-only protein to determine which regions of the protein are exhibiting these differences in exchange. And so, since the gamma subunit is not present in the MoFi protein, we can't really compare the deuterium incorporation between the two proteins there. So I just left it here in green so you can see it and know what's going on with it. So the colors, red indicates an increase in exchange in the iron-only protein, and blue represents a decrease exchange in the iron-only protein. And so the first area that really lit up was this blue region right here, which is adjacent to the gamma subunit. And the fact that we're seeing such a large decrease in exchange in the iron-only protein tells us that the MoFi protein is exchanging a lot there. And so this is consistent with protein-protein interactions, stabilizing this region, preventing exchange. And then another area of interest we saw was around the P cluster and the Phi-Phi-Co, where we're seeing these large areas of increased exchange, suggesting that the iron-only protein is much more dynamic than the MoFi protein. So we were really curious about this and what it means, and wanted to connect it back to the actual mechanism of nitrogen reduction. So we decided to dig back into our cross-linking and HDXMS data. So for cross-linking, we looked for extra long crosslinks with a greater than 30 angstrom cutoff between the gamma subunit and the alpha subunit. And these crosslinks were originally kind of disregarded because they did not fit the distance constraints based on the homology model. However, if we altered the structure of the gamma subunit, or the conformation of the gamma subunit, seen here in yellow, red, and green, we can see that the crosslinks here in black are the distance constraints that become satisfied. So this told us that the structure of the gamma subunit and the gamma alpha subunit interactions are very dynamic, and there's multiple conformations. And then to go back to the hydrogen to deuterium exchange data, I looked for bimodal peptides. And so bimodal peptides indicate multiple conformations of a protein present in solution, and so those peptides are undergoing different exchange rates simultaneously. So it can just essentially causes a widening of the isotope distribution peak. And so two peptides identified were this alpha 221 to 232, and this alpha 93 to 114, shown here in pink. And while these peptides don't interact directly with the cofactors of the nitrogenase, or of the iron-only protein, they do form the surface around the alpha subunit that interacts with the beta subunits, both at the heterotrimer on one side and also the C-terminus of this beta subunit right here that forms the heterotrimer on the other side. So we're really curious about what these different conformations mean in solution. We really wanted to fully understand this. Thankfully, around the same time, our collaborators at Lance Seafeld's lab down at Utah State University published this paper looking at the regulation of electron transfer through asymmetric protein conformations. And what they found is during their pre-steady state kinetic measurements of the iron protein and the MoFi protein, they saw that the two halves of the MoFi protein don't undergo the iron protein cycle independently, which was previously thought. Instead, they hypothesized the electron transfer in one half induces an allosteric conformational change and suppresses electron transfer in the other half. And so essentially, the MoFi protein is going, one half of the MoFi protein is turned on to do the iron protein cycle, and the other half is off. And what they did is they brought in Simone from who I've also worked with, who specializes in mode analysis, a type of protein dynamics calculation. And then, so normal mode analysis determines the amino acid displacement of each of the amino acid residues within the complex. And we can then map those displacement vectors, so shown here as red arrows on the complex, And we see that there's this kind of rocking motion going on between the two halves of the molybdenum iron protein. And then we can generate this covariance matrix here to actually look at the specific relationship between individual amino acid residues with the rest of the complex. And so we were really inspired by this idea. We wanted to incorporate it into our study of the iron-only protein. So we did. And so to kind of give you a little background on normal mode analysis and how to look at the covariance matrix. So the matrix shown here on the right shows the amino acid displacements in the iron-only protein. So to keep it simple, I only showed the alpha-1 subunit on the x-axis, which I've designated as here in the actual complex on the left. And then we compare each amino acid residue to the displacement of each amino acid residue to the displacement of the residues in the rest of the complex. So alpha-1, beta-1, gamma-1, alpha-2, beta-2, gamma-2. And so red regions will indicate an in-phase or correlated displacement. And then blue regions indicate an out-of-phase displacement or an anti-correlated motion. And so by looking at residues that show similar trends, we can potentially start to identify potential pathways of communication throughout the complex. So for instance, we can look at this yellow box I've highlighted here. Starting with the alpha-1 comparison, we see that there's a lot of correlation going on, as well as some anti-correlation. For the beta-1, we see that there's high amounts of correlation. Gamma-1, we only see a little correlation. Alpha-2, we see primarily anti-correlation. And then beta-2, we see more correlation, and then gamma-2, anti-correlation. So to really understand what all this means in terms of the full complex, we need to dive right in and look at the full covariance matrix. So again, this is just the expanded version of the one I showed on the last slide. The x-axis is the amino acid position for the alpha-1, beta-1, gamma-1, alpha-2, beta-2, gamma-2 subunits. And then the y-axis are the same subunits. And so if we follow the diagonal through, this just indicates that each of the subunits are essentially highly correlated with the amino acid residues with themselves. So for instance, if we look at residue 100 in alpha-1, we look at residue 100 here, and we'll see the highest correlation. So then we can start to look at the off-diagonal axis to really identify potential pathways of communication. So for instance, looking at this yellow box again, starting at alpha-1, we see high correlation. Going up to beta-1, we see primarily anti-correlation. And then looking at gamma-1, we see high correlation again. And then looking at alpha-2, a lot of anti-correlation, beta-2 correlation, and then gamma-2 anti-correlation. And so this tells us that a change in the amino acid position or a displacement of the amino acid residue in this region of the alpha-1 subunit causes the beta-1, alpha-1, and gamma-2 subunits to undergo an out-of-phase displacement. And so the movements are anti-correlated. Whereas in the alpha-1, gamma-1, beta-2, we see correlation. So there's some amino acid residues are moving in a similar manner. So this started to really reveal to us that the iron-only protein, similar to the MOP protein, is adopting asymmetric conformations. So each half of the complex experiences different conformations at any given time. And so again, we can also look at the yellow box on alpha-2 and see a similar pattern, just essentially the reverse. So alpha-2 is anti-correlated with alpha-1 and so on. And then we can also look at the green box, looking at the beta subunit. And we see kind of a 50-50 mixture of anti-correlation and correlation with alpha-1, primarily correlation with beta-1, anti-correlation with gamma-1, another 50-50-ish with alpha-2 of anti-correlation correlation, correlation with the beta-2, and then also anti-correlation. So what's interesting about this is, I'm going to jump back a slide, is so the beta interactions form this interface right here at this middle of the complex, connecting essentially the two halves. So the fact that we're seeing high correlation between these two halves suggests that a conformation of one is going to cause an in-phase conformational change with the other, so an in-phase amino acid displacement. So this really helped us determine that the iron-only protein is likely undergoing asymmetric conformations. But something we really wanted to understand was what's the role of the gamma subunit with the iron-only protein. And so we decided to take a closer look at the gamma subunit covariance matrix. Here we have gamma-2 up at the top and gamma-1 at the bottom. And so each of the lines on here indicate different points of interest within the iron-only protein complex. So yellow indicates contacts between the gamma subunit and the alpha subunit. Blue represents P-cluster coordination amino acid residues, and then purple, or pink, sorry, change the color. Pink represents contacts between the alpha protein, alpha subunit, and the iron protein. And finally, green are residues involved in the beta-beta interface. so following along with the yellow lines from the gamma subunit, we see that specifically right here, it crosses and has high correlation with the alpha-1 residues involved in P cluster coordination in iron protein interactions. And then we can see with the beta-1, there's a lot of anti-correlation going on. Alpha-2, there's some correlation going on. And then beta-2, we see more correlation relative to the beta-1 at the beta-beta interface. So this really helped us put together this idea that the asymmetry we're starting to see in the iron-only protein isn't just alpha-1, beta-1, gamma-1 versus alpha-1, beta, or alpha-2, beta-2, gamma-2. Instead, it's actually alpha-1, beta-2, gamma-1, and then alpha-2, beta-1, gamma-2. So that kind of goes back to the interactions at the half of the heterotrimers, likely to coordinate the conformational changes between the two halves, which also helped us really start to learn that the iron-only protein also might experience a negative cooperativity that was originally seen in the MoFi protein. So, to wrap up this part, iron protein has a more dynamic structure than the MoFi protein. Cross-linking HDX and normal mode analysis reveal the dynamics are related to asymmetric conformations in the alpha, beta, gamma subunits, or trimers, excuse me. Normal mode analysis revealed a potential role of the gamma subunit in N2 reduction, specifically based on the in-phase displacements we see in the covariance matrix. We think that the gamma subunit is likely related to regulating of iron protein binding and influencing the protein environment of the P cluster. And so finally, asymmetry in the iron-only protein indicates negative cooperativity is present in all the nitrogenases. And so this really, there's another way to put it, I thought it was really cool. So, but, you know, I wanted to take this another step further and go back to this mechanism of negative cooperativity, which is a form of allostery, which I've already shown we can look at with HDXMS. And so I wanted to revisit the MoFi protein, and specifically with HDXMS, to reveal biophysical evidence of negative cooperativity during the catalytic iron protein cycle. And so the way I accomplished this was by using a quenchable apparatus in order to do the mixing of the deuterated and non-deuterated solvents. So with this, we were able to potentially achieve our goal of obtaining biophysical evidence of negative cooperativity. And with the quench flow, we can perform rapid deuteration measurements in order to look at the protein dynamics of the MoFi protein on the catalytic time scale, specifically during the different steps of the iron protein cycle. And so to quickly tell you the setup, syringe A contained the MoFi protein and non-deuterated buffer. Syringe B contained deuterated buffer with and without iron protein and ATP. And then syringe C contains the formic acid, which is the quench solution. And so A and B would rapidly mix for a certain amount of time designated by the sample loop. And after they mix for that amount of time, they would come into contact with the quench solution, and then we would be able to collect the aliquots and then analyze them with mass spectrometry. And so the three conditions I tested with this were MoFi, so just the MoFi protein, MoFi to iron protein 1:1, so MoFi to iron protein mixed together in a 1:1 ratio. So in theory, there should only be one iron protein bound for every MoFi protein. And then MoFi to iron protein 1:2, mixed in a 1:2 ratio, so there should be 2 iron proteins for every MoFi protein. So the first step in the analysis of this was to really determine, can we see the binding effects of the iron protein with the rest of the MoFi protein? And the way I accomplished that was to actually generate these butterfly plots comparing the percent deuterium incorporated in the MoFi and MoFi to iron protein 1:1 condition, shown here on the graph on the left, and the MoFi to iron protein, MoFi and MoFi to iron protein 1:2 conditions on the right at the 10, 80 millisecond, 2 minute, and 1 hour time points. So 10 millisecond is going to be the blue trace, 80 milliseconds the gold, 2 minutes the purple, and then 1 hour the red. And so, I realize this is a lot to take in, so I'm going to break it down for you. So each graph shows the x-axis with the peptides identified during the HDXMS experiment. And then the y-axis represents the percent deuterium incorporated in the MoFi condition for the positive values, and then the MoFi to iron protein 1:1 condition for the negative values on the left, and MoFi to iron protein 1:2 condition for the negative values on the right. So looking at this, we can go through and look at these different regions of the MoFi protein and see, can we detect noticeable differences in the exchange rate when one equivalent of iron protein is bound, or when 2 equivalents of iron protein are bound. And so instead of rattling off every single one of these for you, I'm just going to show you some specific ones. So the first is this beta 183 to 192. So we see in the top half here, the Mo-Phi condition incorporates the most deuterium by the 1 hour time point, and it also has a fairly steady rate of exchange. In the Mo-Phi iron protein 1 condition, we see a slight decrease in the overall amount of exchange. And we also see the earlier time points are much more closer together, indicating that there's possibly a slower rate of exchange going on. And looking at the Mo-Phi to iron protein 1 to 2 condition, we can see that the overall deuterium incorporated by 1 hour is much less, and we're seeing a similar clustering of the earlier time points with the Mo-Phi iron protein 1 to 1 condition. So the main takeaway from this peptide is that when 1 iron protein is present, we can see a noticeable difference in the amount of deuterium incorporated. And when there are 2 iron proteins present, we can see another noticeable difference. And so this helps us confirm that we can detect when 1 iron protein is bound with the Mo-Phi complex and when 2 iron proteins are bound. But the cool stuff doesn't stop there. So next, this alpha 39 to 54 region, we see a slightly different behavior than the last one. First, in the Mo-Phi condition, we see again a kind of steady rate of increase with the most incorporated by 1 hour in the Mo-Phi condition. But however, in the Mo-Phi to iron protein 1 to 1 condition, we see that the earlier time points are spread farther apart, indicating a slightly faster rate of exchange at each time point, and then it overall incorporates less after 1 hour. And then in the Mo-Phi to iron protein 1 to 2, we see a similar, exchange rate in the 10 to 80 millisecond time points, and then the 2 minute and 1 hour time points are similar to the Mo-Phi condition. So this tells us that when only one equivalent of iron protein is bound, we're seeing actually an increase in exchange. And this starts to kind of tell us that maybe this is going back to the negative cooperativity behavior and the asymmetry of the Mo-Phi protein. And so finally, another region that kind of confirmed this is beta 114 to 128, where we see a very slow amount of deuterium incorporation in the Mo-Phi condition. We see the Mo-Phi to iron protein 1 to 1 condition exchange very quickly. And then in the Mo-Phi to iron protein 1 to 2 condition, we also see a much slower rate of exchange. So, looking at these butterfly plots really convinced me that there's likely some, if not directly related asymmetry, there's just something interesting going on with the Mo-Phi iron protein 1 to 1 condition, specifically the dynamics and the accessibility to exchange in the Mo-Phi protein. And so to kind of connect this back to the previous study of the iron-only protein, I looked for more bimodal peptides in the Mo-Phi protein. So to remind you, bimodal behavior, or bimodal exchange, indicates multiple exchange rates. So the protein is adopting 2 different conformations, causing 2 different amounts of exchange. And these isotope distributions will overlap, causing a widening. And so I identified these 3 regions as bimodal, with bimodal behavior present only at 10 milliseconds in the Mo-Phi iron protein 1 to 1 condition. And so the first region is this alpha 102 to 117, which forms part of the surface of the alpha subunit near the P cluster, and includes this 308-104 residue, which has been implicated to be involved in a potential pathway substrate binding that allows nitrogen to travel to the FeMo-Co. Next is this alpha 208 to 230 region. And so you can see here in this inset, it interacts directly with the FeMo-Co through this tyrosine 229 residue. And then finally, alpha 186 to 200 also forms part of the protein environment of the FeMo-Co and includes this histidine 195, which has been shown through mutagenesis studies to be crucial for nitrogen reduction. So the fact that we're seeing all this bimodal behavior around critical sites of nitrogen reduction really helps us confirm that there's asymmetry going on and potentially negative cooperativity. But I didn't set out this experiment just to look for, are we seeing asymmetry in the Mo-Phi protein with Hg XMS? So I wanted to connect it back to the structural asymmetry originally observed from our collaborators using normal mode analysis. And so I did this by looking at the dynamics of the Mo-Phi, or the amount of exchange in the Mo-Phi protein on the catalytic timescale, specifically the iron protein cycle. So I was able to do this by using the rate of electron transfer, the rate of the different steps in the iron protein cycle, to determine the approximate timescale they would occur on. For instance, with electron transfer, rate constant is 140 per seconds, which comes out to about 7 milliseconds. And then for ATP hydrolysis, about 14 milliseconds, and then inorganic phosphate release at 62 milliseconds. So, I looked at the millisecond time points collected with quenchable HGXMS to really characterize this. And so, I have a movie to show this, which I'm very proud of, but before I hit play, I want to make sure everybody's on the same page to know how to read or look at the heat maps. And so, here are the structural heat maps of the MoFi protein with the MoFi to iron protein 1:1 versus MoFi at the top, and MoFi to iron protein 1:2 versus MoFi at the bottom. And again, when we see red regions, that indicates an increase in exchange in the iron protein bound condition, and blue regions indicate a decrease in exchange in the iron protein bound conditions. I'll take a quick swig of water. Okay, so we start off with our ATP bound iron protein, MoFi protein complex. So after electron transfer, at 10 milliseconds, we can see the state of the MoFi protein dynamics after electron transfer. And so what we see is that overall they indicate similar patterns of exchange based on the largely blue regions indicating protection when iron protein is bound. However, what we start to see in the MoFi to iron protein 1:1 condition are several regions indicating an increase in exchange, specifically around the iron protein binding site, P cluster, and the Phi-MoCo. So to take a closer look at that, again, increased exchange at the iron protein binding site, P cluster, and the Phi-MoCo. And so then the next step is looking at the 30 millisecond time point, which captures the dynamics after ATP hydrolysis. And again, overall, we don't see much of a difference compared to the 10 millisecond time point. We're still seeing overall protection in both of the conditions, and the regions indicating an increase in exchange in the MoFi to iron protein 1:1 condition are still showing that behavior. However, something interesting is that around the Phi-MoCo, which I'll show another zoom in just a second, shows slightly less exchange, indicating it becomes more stabilized. This is likely when electron transfer initiates. We see this decrease around here, suggesting that essentially the nitrogen substrate channel closes. And then at the 60 millisecond, right before inorganic phosphate release, what we see is actually a change in between both of the conditions, primarily at the beta-beta interface in the middle, which you can't see now because the time is poorly. Now that we're here, we can actually see an increase in exchange at the C-terminus of each of the beta subunits, indicating that there's some kind of crosstalk going on between the two halves of the MoFi protein. I'll show that again in just a second. So here are the regions I'm indicating. And you can see you can almost follow increases in exchange from one half of the complex to the other. So this started to tell us that maybe this is a potential way to send a signal across the two paths. This is actually confirmed by looking at 80 milliseconds after inorganic phosphate release, because in the MoFi to iron protein 1:2 condition, this behavior isn't there anymore. It's back down to blue, indicating a decrease in exchange, meaning it's stabilized.
Look, can I ask a question on the 1:1 complex? Wouldn't you expect it to have an equilibrium where the iron protein would dissociate and bind to the other side?
Yes.
So would that be, okay, go ahead.
No, yeah, so, it's a great question.
It's also a population average, right?
Right, exactly.
So you're mixing the bound and the unbound conformations together.
Right. So that's something we considered when we first started analyzing this data is, are we really seeing just one iron protein bound to the complex in the MoFi iron protein 1:1 condition? Are we seeing some with two bound, or are we seeing some with none bound?
Right.
And so that kind of goes back to the butterfly plots, where based on the differences we see between each of the conditions, we can say that the majority of the population, at least for the MoFi iron protein 1:1 condition, is the one iron protein bound relative to, does that make sense? Kind of.
Could you distinguish that with cross-linking, or is that too crude?
I think we could. Potentially, it might be difficult to get it on the time scale that's shown here, but I think that's a good way to see, are we...
You have 2 to 1 and some and 0.
Exactly. And something else we've considered is essentially redoing this experiment, but using iron protein, or deuterating the iron protein at these time points to again see if we're kind of getting the other half of that. So, this pathway of communication across the two halves, and so we see this behavior persist at the beta-beta interface in the MoFi iron protein 1:1 condition, but we don't see it in the MoFi iron protein 1:2 condition. So this kind of convinced me that there is a signal going from one half to the other to essentially, after inorganic phosphate release to switch this side of the MOFI protein off, and then make this side available for iron protein, the iron protein cycle. And so to kind of show that on here, we have a release of the inorganic phosphate starting here at the iron protein binding site. We can go through, travel through the P cluster to the C terminus of this beta subunit in one half of the complex. It'll jump across to the other half, and then travel back through to the other side, essentially making the site available for electron transfer. So, kind of summarize all that up. Overall, with bimetal exchange, we were able to determine that there is an asymmetric behavior when iron protein is bound. Quench flow HTXMS showed the dynamics of the MOFI protein on a catalytic timescale. With the MOFI to iron protein 1-to-1 conditions unique exchange pattern, revealing potential pathway communication connecting the iron protein binding sites. And finally, this relates back to allostery, or native cooperativity, facilitating the ability of each MOFI protein half to transfer electrons. And so, by suppressing the first electron from P cluster to PMOCO, based on the increase in exchange here, it's likely to cause changing the distance between these two, between the PMOCO and the P cluster, so the P cluster can't do that first electron transfer. At least not at the same rate as it normally would. And then finally, to also prevent energy loss while it's getting ready to reduce nitrogen. And so something else I would really like to do with this study is go back into the normal mode analysis data of the MOFI protein to really further confirm the pathway of allostery, but also to look at the bimodal peptides and some other regions to see what kind of covariance we're seeing in the complex. So, on that note, I'd like to thank my committee, the Mass Spec facility, Jesse, Ganesh, and Jonathan for handling all my minor panics when something goes wrong with the instrument when I was first learning them. Betsy members from the Biological Electron Transfer and Catalysis Research Center, Gertie, who helped prepare the NFM protein, Natasha Pence, who started here at MSU, is now at Washington State University, who helped with a lot of the iron protein. We have Saroj from here at MSU, who helped with the statistical coupling analysis. Then Lance Seefeld's lab, with Hayden and Derek, who helped me with the MOFI protein quench flow experiment. And then all these people from the Botnet lab, who not only helped me do these experiments or learn about these proteins, but also offered some additional support. And then, of course, family stocks. So, on that note, I'll take any questions.
Absolutely beautiful work. I wonder, could you distinguish when you have the 1-to-1 complex, there's a puzzle about whether there's a mixture of 0 to 1 and 2. The ion mobility mass spectrometer, if you cross-link first and then put it through the ion mobility mass spectrometer, I think you could you see that?
I think we definitely could, just based on the collisional cross-section.
Yeah.
It would tell us enough about, in theory, it will tell us enough about whether there is 1 iron protein down or 2 iron protein down.
Or 0 in some
Or
cases.
0 in some cases, but kind of what the major population conditions is.
So, it's just more of a technical question, I guess, because we've talked about this HDX data, you know, in our lab with some stuff, and even in other experiments, you're talking minutes to hours to overnight HDX versus milliseconds.
Yep.
So, what's your level of exchange at those timescales?
So I think the most exchange I see on the millisecond timescale is about 30%.
That's pretty good.
Based on the 80 millisecond time point. I can actually go back to the butterfly plot. It's kind of difficult to see on here, but so yeah, the highest one, like this one right here, is actually about 20% at 10 milliseconds, and then about 40%, right under 40% at 80 milliseconds. It's a little higher over here, but
So there's pretty good confidence in your.
yeah, it works really well.
If nobody has another question, I'll ask her.
Yeah, go ahead.
I'm a little rusty on this normal mode analysis business. I mean, it looks very nice. Has it been presumably worked out in little simpler systems to sort of kind of show that it's giving you good answers, the normal mode analysis?
It has, yes. I can't remember. I have the title of the paper in my head, but because I mainly looked at it for these nitrogen systems.
Sure. Yeah.
Trying to get the background
Just
on it.
wondering what else has been done with it. Has it really been validated, because it looks nice.
It has. They've actually validated it a lot, and I think the main way they did that was actually looking at the protein data bank and looking at the crystal structures in there.